Hopefully. Hi, Elena.
Yeah.
Thank you
Hi.
very
Yes.
much. And yeah, oh great, we can see all the panel now. So
Yeah.
thank you. So we've had a couple of questions in the chat. So I'll kick off with a couple of questions to Steve, please, about implants. So the first one was just a question about if artificial irises had a slightly bigger pupil, would that help overcome some of the problems such as viewing the peripheral retina or viewing the back of the eye afterwards, do you think?
Yeah, potentially, yes. I mean, they're not, the flexible ones are quite easy to modify. You can actually, if you wish, enlarge the pupil with like a skin punch trephine. You can just make it bigger if you wanted to do so, but you have to do that before it's implanted. You can't make it bigger once it's implanted without taking it out again and putting in another one.
Okay. And the other question about implants was, do you think the implants can increase the risk or worsen keratopathy or limbal stem cell function?
Surface function, no. I mean, on the talks, there have been several illustrations of people who've had corneal transplantation. So definitely the implants can affect the corneal endothelium and lead to decompensation. I think if people didn't do intraocular surgery, the risks of corneal decompensation, as opposed to stem cell failure, would actually be very low in aniridia. I think most of it is surgically precipitated.
Brilliant, thanks. Which I think speaks to what I really noticed what Keith said, his mother said, about don't let them start messing with your eyes, i.e. operation. I couldn't agree more. If you don't need an operation, don't have one. Ellen, I think you had a question for Tony.
Yes. Tony, could it be possible that your patient with aniridia and hearing loss has two independent conditions, since hearing loss is quite a common disease?
Yeah, basically the thing about Eva's research is that it was looking at central auditory processing problems. So in her studies, all of the patients with aniridia that were studied, the adults and the children, had normal hearing when you tested each ear independently. You did audiometry in the normal way. So they didn't have a normal hearing problem. But what was abnormal was that tests that are designed to look at how you process hearing once it gets to the brain level were abnormal in most patients with aniridia. And in fact, I'm not a hearing expert, as you know, but there's been another recent study from the States where they've looked at central processing by looking at hearing, the EEG, putting electrodes on the scalp and measuring hearing response to the brain, which are also abnormal in aniridia. So I think there is a very specific auditory processing problem, which manifests itself in children and adults not hearing very well in crowded situations or where there's a lot of competing noise. And if they just go to their normal hearing clinic, it wouldn't be picked up. So it was just an illustration that there are subtle wiring problems in the brain if you have an abnormality of the PAX6 gene.
Excellent. Thank you, Tony. And I've got a question for Dominique from Linda in the chat, which is, Dominique, is there a known connection between aniridia and Ehlers-Danlos syndrome?
Well, that's a very good question because I work on both syndromes, and at the time I didn't find any connection for that. And as you see, there is also other syndromes involved in those children. But as I have a very large cohort, because 342 is quite a large cohort, there may be an association, but a hazard is association. So at the time, I have no answer to relate both syndromes.
Okay. Thank you. And one of the questions is, Dominic, if you're okay to take this, and if not, others could chip in from the chat. I'm not sure who it's from, though. If diabetes occurs in a patient with aniridia, what sort of age are they generally affected?
So we are working on that for the court, and usually it's more about 30 to 40 years old that it can occur, but we have some very juvenile diabetes like before 20 years old. So I think we could be aware of it. It's very complicated to ask for glycemia every year, but to be aware to the parents to say that they can just follow, for instance, the water consumption and any sign that can be different in the life of the child. But usually it's later. It's later that.
Okay, that's great. Thank you. Now, I think, Sarge, I think you had a question for Jay, well, and Helena, who's covering for Jay. So do you want to fire away with that?
Thanks, Mel. Helena, Jay was mentioning about seeing patients who have nystagmus and then have genetic mutation screening done, and you find a PAX6 mutation. I've seen several patients who have actually a full iris and pretty much nothing to see that's abnormal on the iris, and yet they have significant stem cell failure, and that's the only thing that you'll see. You do their mutation screening and they have PAX6. So do you see patients occasionally that have nystagmus and little else, normal iris, and they end up having a PAX6 mutation?
Yeah, we get them. What we might only detect is subtle foveal hypoplasia when we can eventually get the children on an OCT scanner. So often we'll pick that up on genetic screening a little bit unexpectedly. We might have thought they may have been albinism, but it turns out to be PAX6.
And have you looked to see if there's any specific mutation that results in that? Because what interests me the most is, are the patients that I see with corneal problems but nothing else and normal iris, and the patients that you see that have nystagmus but a normal iris, do they have a specifically different mutation to the vast majority of other patients that have
That's a really interesting question.
Because I think that might actually hold a key to some of the issues in development that we see in aniridia.
Yeah, we haven't specifically looked at specific mutations or done the functional studies that would need to be done to prove this sort of thing, but it would be really interesting to do, and maybe, gather groups of these cohorts and actually look at it and see, yes, this specific mutation really affects corneal stem cells. This specific mutation causes more of a retinal phenotype, or this one, you're going to get glaucoma. It would be nice to actually truly understand that. I think maybe Dominique might have more information on that, because it sounds like they have a huge genetic database.
Well, no, I don't have anything to add for that.
But Maria, no, I know you do, and I think you're thinking what I'm thinking. So there is something we can say about genotype, phenotype for these, if you like, slightly different presentations. Do you want to comment?
Yeah, so from our cohort at Moorfields, it's the patients that have the missense mutations that usually have the milder phenotype where the iris is mostly intact, but you get nystagmus and a degree of foveal hypoplasia. And there is that issue about whether or not there is limbal stem cell deficiency also. But it's usually the missense mutations. The caveat is obviously there is a particular missense mutation in a SOX2 binding domain, which causes a more severe phenotype. But if you exclude those particular mutations, it's the missense group that will give you that presentation.
Maria, could you just explain in a lay manner what's a missense mutation and how that differs from other mutations, please?
Yeah, so we have various different types of mutations. So some mutations, it can involve the whole of the PAX6 gene being cut out of the DNA. So we call that deleted. And sometimes certain parts of the PAX6 gene can be deleted. These are all classified as deletions. And so we don't have enough of the PAX6 gene present and working. Sometimes we just have a single letter genetic change, like a spelling mistake in our genetic code within our PAX6 gene. And some of those can just change the instructions. So instead of a subtype of the protein, changing it to a different type of subtype. And that's a missense mutation. So for those who need a bit more information, we're talking about amino acids. Amino acids are small subunits that make a protein. And so with a missense, it's a single change that introduces a different amino acid. There are other spelling mistakes that can actually introduce an abnormal stop signal. And that when your cells are reading your gene, it essentially just stops producing it. It's like a red light. And so you don't get, again, PAX6 protein. And these can arise from, a few types of different mutations, so called frame shift mutations or nonsense mutations. So that's a kind of general overview of different types.
Thanks. And Dominique, I think you were going to come back to us.
Yes, just very shortly. Yes, of course, we know that missense mutation can do a milder phenotype, but in my experience, some are missense and they have sometimes a very severe phenotype. So we must be aware of it. And the second point I observe is really the penetration of the phenotype. Because one of my presentation, the mother was just identified as a coloboma of the iris, and the child had a complete and severe features of the congenital aniridia. So we must be aware of sometimes we have some. I think we have some principles that are not exactly the same when we observe a large cohort. Just my experience.
And Maria, I think you had another question regarding this sort of area for Dominique.
Yeah, it's a question for Dominique, Steve, and Sarge, actually, for those of you who see the cataracts. Dominique, you really nicely showed that picture of the cartwheel cataract. I've never seen that before, but a lot of my patients normally have extraction before I do see them. But is that pathognomonic, and what are the different other types of aniridia type of cataracts that we see?
Yes, I think we are about to publish that, and it's really, to my thinking, it's when you see the cataract, you say, okay, that's a cartwheel, okay, that's a PAX6 mutation, which is really unusual to just look at the lens and say, I know what kind of gene is involved. And of course, not all of them have the cartwheel cataract, but we can see the evolution, so that's really nice. And usually, when the cataract is done, and it was before that, we didn't took any photos, and I take photos of every child and every adult that I can observe, and that's why we observe that, and we observe also the evolution. So I think it's quite characteristic because I didn't see in another mutation. And of course, in like other mutation, CYP1B1 and the other FOXC3, there is no this kind of cataract.
I think the other thing that I would add to that is, certainly from having operated on a few aniridia cataracts now, is that the anterior capsule appears very abnormal often and won't behave like a normal capsule would during surgery. And there clearly is something, and it's not surprising, that there is something going on in the front of the lens as well, and whether or not it's the capsule that causes the cortical changes anteriorly, or whether it's the other way around, is difficult to tell. But there is clearly something going on, and it's important for us to know, because that will help us determine how easy or difficult cataract surgery will be, and hence the outcomes.
Steve, did you have your hand up there?
Yeah, it's just a quick question for Dominique, whether there is any genotype that correlates with the sort of inferior zonule absence or deficiency, or inferior lens coloboma, because that's a practical, has practical implications for the long-term survival or success of cataract surgery, is lens dislocation.
That's a very good question, because I'm collecting the data, because it takes a long time to look at the genotype and to make the I mean, the relationship between. And I don't think at the time we have a real specific genotype for this kind of coloboma, because it's really very unusual. So it's very difficult to collect a lot. I don't know if some of you have the same cases as I just, or similar cases, because it's never the same. I don't know if you had that. It's quite complicated also, because those lens coloboma, when we operate them, they have more glaucoma, and I don't know why. So if some of you have had some, just let me know.
Helena, do you want to ask the next question?
Yes, I think this is to the entire panel. Is there any known connections between PAX6 and mental illness, even in those without aniridia? Both Dominique and Maria want to answer that. So we'll start with Dominique.
No, just Maria first, and I will.
Oh, Maria. Okay, Maria. Okay.
You said first.
So you're going to hear my presentation in the next session, but one thing that we have found is that serotonin levels are significantly reduced in the aniridia patients compared to healthy age-matched controls. And obviously, low serotonin levels are linked to mental health issues like depression. We take serotonin in reuptake inhibitors, etc. As a treatment for that. So I think this is an area that needs further exploration. We're not sure yet if these mental health issues are linked to the PAX6 mutation directly and the molecular results, or is it linked to the sight loss and the effects on an individual themselves? But certainly from a metabolomic kind of viewpoint, there is certainly indications that there may be some possible molecular link with aniridia.
That's
Great.
interesting.
Helena, I think we're going to have to stop now. So do you want to round up, and then I think it's hand back to Dave after that.
Okay, so that was a brilliant session, I think. Some very interesting discussions and very informative talks from all the speakers. I think we can take a lot away from this. We're learning so much more about the genetics. We're learning about the importance of communication, and treatments seem to be evolving. So I'd say there's a lot of hope on the horizon for aniridia and research and the future of vision, and indeed things like mental health for our aniridia patients.